Sorry.
Alright. Hi everyone. So you're probably wondering why we're standing up here, and to be perfectly honest, we're not entirely sure either.
Yeah, so I just want to let you all know that if you have any questions at all, don't be afraid to stop me. And it's going to be a great intro for everyone. And if you don't understand something, just let me know.
Right. So, last week, this guy, James, messaged both of us. And we don't really know who this, like, John person is.
Yeah, no
So-
idea about this Jasper fellow.
So, yeah, we were pretty confused, the two of us.
Yeah, we were pretty confused, and you know, we did what any good millennial should do, which is Facebook stalk this James guy, and we found some pretty interesting stuff.
So we found some photos. More photos.
We started to sense a trend. Not the mustache, but the apparent widely different hairstyles used by one individual. There's a lot.
And we were even confused because this Josh person, we have no idea who this person is. But they were even like here at Harvard with their
Whoa.
mustache and like,
Wait, I did a project with him?
yeah.
Oh my gosh, who is James?
I don't no idea.
Oh, John, such a mystery.
Yeah. So,
We continued our Facebook search to sort of get to the bottom of who this character was. And he claims to have gone to Texas to do cancer research, but we all know that's probably a weird alibi, right? I don't know about that. So then we decided to do an actual publication search to see if, you know, he actually had any real backup to explain his And this is all we found. So, you know, this made us a little suspicious. We sort of formulated a hypothesis.
so we know that this person keeps changing their appearance, keeps changing their name,
He loves to sew.
loves to sew, and seems to have spent 3 years somewhere doing
Somewhere.
something.
He has a pretty good alibi, according to this science article.
But we came up with the only reasonable conclusion,
Yeah.
which is that Jimmy is a superhero. And so we did a little bit of searching, sleuthing.
Yeah, we were like, what would his superhero be? You know, like, what would he, you know, want to cultivate before he came to MCO to unleash it upon, you know, this research community? What would his ideal superpower be? And we concluded that the only, you know, real superpower that we could think of is that he's able to eject pipette tips only using his mind. So that's Jim for you. And with that, I think we'd just like to welcome Joshua up to the stage. Yeah. Love Joshua.
All right, thank you for that introduction. Can everyone in the back hear me okay? All right, good. Now, I'd like to start my presentation on axolotls and on limb regeneration. My name is actually Jclub Austin-Breneman, and I'm an MCO graduate student. And when I go to a new locale, a new venue, and I talk about science, the first thing that I think of is how do I make my science understood to people who may not be in that field? So naturally, what I think is, how could I explain this to my 5-year-old niece? So what I did today is I brought in my favorite children's book from when I was growing up, Allie the Axolotl and the Mystery of Her Missing Limb. So axolotls are a type of Mexican salamander that lives in a lake called Lake Xochimilco. Now axolotl were very happy and healthy in this lake, but now the lake is under Mexico City, and so there is no lake anymore. And the main place that you find axolotl are in the tanks of research scientists and pet owners with expensive tastes. So the main character of this story, and my favorite children's story, is Allie the axolotl. And at the start of our story, we find that Allie the axolotl has lost a limb. And she doesn't know what's going to happen to her. Now, losing a limb is pretty common if you're an axolotl in a research laboratory. But this is the first time for Allie the axolotl. And she doesn't know what's going to happen. So she finds that after a few days, a bump has grown where her arm used to be. Now, she has no idea what's going on, so she does what I do when I have no idea what's going on. She asks a professor for help. So Professor Axolotl is a renowned research scientist in the axolotl field and knows all about axolotl limb regeneration. And so decides to explain to Lucky Allie the axolotl all about limb regeneration. Now the process is fairly complex, but can be boiled down to a few steps. First, axolotls can have a perfectly healthy, fully developed arm that, through some mishap or other accident, is amputated. After a few days, the axolotl's limb will grow what is known as a blastema. The blastema is a mass of cells that, together with the axolotl's body, will regenerate the entire limb that was missing. This limb is almost identical to the limb that was missing, to the limb that was originally on their arm. A little history on the theory behind blastemas. That blastemas are thought to be homogeneous, a mass of cells that were all identical and all capable of regenerating these cell types in the newly regenerated arm. Now this is very important because if that's true, it means that there's a single cell type, a pluripotent cell, that is capable of regenerating many, many different cell types. And if we humans could identify the factors that make these blastema cells so special, then we might be able to use that for people who have lost arms or legs and help them recover. So this is a very important field of study. The reason why blastemas were thought to be homogeneous is due to a series of experiments that used dye labeling. So in these experiments, cells such as muscle cells were labeled with a dye. The muscle cells were transplanted into the arm of an axolotl. The arm is amputated. And when it regenerated, they found that they found dye-labeled cells in all of the different cell types making up this new arm. So they found from these dye-labeled muscle cells, more muscle, cartilage, dermis, nerve cells, all the different kinds of cells containing this dye. So that's pretty good evidence, but not concrete. And that brings us to today's paper, Cells Keep a Memory of Their Tissue of Origin During Axolotl Limb Regeneration, by Martin Kragel and his group. Kragel and his group had some problems with the dye labeling experiments. One thing, a paper in 2006 showed that if you inject neural type cells, if you transplant neural type cells into the blastema of a regenerating tail, you can find those dye labeling in all of the cells in the nerves of that regenerated tail. But if you do the same thing with dead cells that are dye labeled, you still find dye in all of the regenerated nerve cells. So clearly there's something not quite right about using dye labeling for these experiments. So Kragel and his group did something a little different that may be more common to us now. They used axolotls that endogenously expressed GFP. So the limbs of these axolotls and all their cells are expressing GFP. Kragel took these GFP positive cells from the dermis and transplanted them into the dermis of a GFP negative axolotl. he allowed them to heal and then amputated the limb. A blastema formed, which incorporated some of these GFP-positive cells, clearly indicating that these dermal cells are contributing to the blastema. But then when the entire limb had regenerated, and he could look at where the GFP marker was, he found that it wasn't in all of the cell types. It was in dermis, cartilage, in some Schwann cells, and in interstitial fibroblasts in the muscle, but not all of the cell types. Now, this is contrary to what you would have expected if the blastema was homogeneous. You would expect that if all the cells are the same, you will see GFP-positive cells in all of these different cell types. A second experiment looked at cartilage. Using the same process, transplantation of cartilage from a GFP-positive axolotl into a GFP-negative axolotl showed similar results in terms of blastema. The blastema did incorporate GFP-positive cells. And then again, when the limb was regenerated, not all of the cell types contain GFP marker. In fact, only cartilage contained GFP marker. So what does this mean? Well, it suggests that some cell types are lineage restricted. Epidermis only went on to make certain cell types, like epidermis and cartilage. And cartilage cells only ever went on to make cartilage in the newly regenerated limb. So this idea that the blastema only has one type of cell that goes on to make up all the other types of cells is clearly not the case. But this evidence wasn't quite concrete enough for Trowell and his group. Looking at it, they decided that they needed cleaner experiments. That, for example, if you look at the dermal transplant, the section that they used to transplant contained dermal cells, but also epidermis, some Schwann cells, some blood vessels that may have contributed to these other labelings that they saw. So they used an even more refined technique to label embryos. So they started with an embryo of an axolotl at about 60 hours post-fertilization. At this stage, they collected a part of the pre-Semitic mesoderm and transplanted it to an axolotl of the same age that was GFP negative. This section of mesoderm only went on to label muscle in the limb when the axolotl was fully grown. So here, the muscle is clearly labeled and no other cell types are labeled. After amputation, the blastema again incorporated GFP-positive cells. And as you might expect now, because this paper is very important, they saw that muscle was the only cell type to express GFP, suggesting that the GFP the muscle cells only went on to make more muscle cells. To confirm with another cell type, they looked at labeling only Schwann cells. So Schwann cells, as you may or may not know, are the component of nerves that wraps around the axon to form myelinated sheaths. After amputation, again, they had GFP-positive blastema. And instead of labeling muscle or any other cell type, they only labeled Schwann cells. The Schwann cells only went on to make more Schwann cells. So what does this tell us? Again, we can further confirm lineage restriction. Muscle cells only go on to make more muscle cells, and Schwann cells only go on to make more Schwann cells. And so, again, the idea of a homogeneous blastema is further disproved. What conclusions can we draw from this information? Well, for one, we can see that using GFP positive axolotls yields a cleaner and more accurate labeling of individual cell types than the dyes. We also see that there is lineage restriction, that one cell type only goes on to make more of itself. It does not go on to make more of another kind. One exception in this paper being dermis, which contributes to both dermal cells and to cartilage cells. But most importantly, and the reason why I chose this paper, is that this shows conclusively that the blastema is not homogeneous. It is a heterogeneous mixture of cells that have a predetermined fate. But that just wasn't enough for this paper. So they looked at not only cell lineage, but also at position, and whether there's homogeneity in position of the blastemal cells and their origin. So in this experiment, Crowell and his group took cartilage from either the upper arm or from the fingertip of a GFP positive axolotl and transplanted it into the upper arm of GFP negative axolotls. Again, GFP positive blastemas, showing that these cartilage cells were incorporated into the blastema. But when they looked at where GFP was expressed in the regenerated limb, they saw that when cartilage from the upper arm was transplanted, cartilage cells all along the arm past the site of amputation expressed GFP. But from the fingertip cartilage, only fingertip cartilage cells express GFP. None of the cartilage cells from that were proximal to the fingertips express GFP. But in a somewhat interesting twist for this experiment, they looked at the same process using Schwann cells, taking Schwann cells from either the upper arm or the fingertip and transplanting them to the upper arm. And with Schwann cells, you actually see that there's GFP positive Schwann cells all along the regenerated arm, regardless of the site of origin of the transplanted Schwann cells. And that says that there's positional restriction for some cells. Schwann cells do not have positional restriction. Regardless of where they come from, they can go to any site in the newly regenerated arm. Cartilage cells, however, only go to sites that are more distal to the site of amputation. So the site of amputation is here. Cartilage cells will only go from the green, from here on, or from the fingertip to the fingertip. This study came out in 2009, and since then there have been further techniques that have been developed to help determine cell lineage and do cell tracing. One such technique was developed here at Harvard and is known as Brainbow, which I'm sure you all have heard about. Brainbow allows you to label individual cells with different colors and track them across development. Here, this group used Brainbow to look at a developing fingertip. They marked individual cells in the fingertip of different cell types and tracked where those cells went to, which cells were generated from those marked cells. And they found that, again, cells of one type only go on to make more cells of that type. So now that Ali the axolotl has sat through Professor Axolotl's lecture, she does what I always do after a lecture, and she thanks the professor. And I would like to thank my axolotl professors, Michelle and Monsi, and also Yuchun for helping me develop my presentation. So thank you, and I'll take any questions.
Can limbs be regenerated at any stage in life, so whether they're very young or very old?
Yep. So axolotls actually are a pretty interesting creature in that they stay in a permanently juvenile state that is able to reach sexual maturity. But yes, they can regenerate at any stage in their life, and there's actually been a lot of studies that look at whether age affects regeneration, and it does a little bit, but what affects it more actually is if you amputate an axolotl limb, let it regenerate, amputate, regenerate, eventually the ability of that limb to regenerate fully is decreased. And so you have, after 8 or 9 regenerations, you start having axolotls that are missing limbs, or that are missing digits, or their bones aren't forming properly. So yeah, so they're capable, but it's not unlimited.
So you drew the blasphemer as being very sort of uniformly mixed, like very well mixed cell types. But the fact that these cell types are lineage specific means that they would then spatially desegregate. Did they check that? Is there spatial segregation?
So there is spatial segregation that we know now. Actually I was reading an interesting paper that electroporated different markers into either the more proximal part of the blastema or the more distal part, and looked at where those parts ended up. It's a not entirely solved question in axolotl blastema regeneration. But yes, there is some spatial heterogeneity, but this paper didn't show that, so I didn't want to draw a conclusion that wasn't there.
Thanks.
Extension of question. So if this paper or the paper study, you can also check, say, for molecular level expression, like some biomarkers that are more specific to the certain types of the cell or the other, or maybe potential RNA expression of the different cells
In the blastema? So they didn't do that in this paper. That's definitely an important question, and one that people are working on. There aren't clear, I know that there aren't clear expression sets for positional identity in the blastema. As for cell type, I'm not sure. They didn't look at that in this paper. Thank you very much.